So welcome to the Roberts lab meeting. Today we're going to have the members of the lab talk about what they're planning on doing this semester.
Hi, I'm Tess. I am a second-year master's student, so I will be finishing this semester, fingers crossed. What I plan on doing is I had previously made a ton of mutant moss. We study cellulose synthase proteins, so I mutated those genes basically. And now this semester I plan on going in and testing through Western blot to see whether or not those genes that I made are actually making the protein. The other thing that I'm going to do is I'm going to further mutate a very specific region in the cellulose synthase protein to see how it functions for the entire protein.
Hi, my name is Mai. I'm a fourth year PhD graduate student. Right now I'm working on seeing where the cellulose synthase genes are expressed. So I'm making reporter gene line constructs. So wherever this gene is expressed, I'll see blue staining when I stain the tissue. And based on where these genes are expressed, I plan to do RT-Q-PCR to see the expression levels. And Western blots to see the actual translation of the proteins as well.
Hi, I'm Bailey. I'm an undergrad here. I just started working in the lab, and right now they're having me pretty much look at the moss that they have attempted to mutate and determine whether they have successfully mutated them. And so I do that by PCR and gel electrophoresis.
Hi, I'm Jason. I'm an undergraduate here at URI, and this semester I'm working with Dr. Roberts to produce a new version of the SSA5 knockout. The SSA5 knockout is characterized by the lack of a gametophore, but sometimes it errors and a gametophore occurs anyway, so ideally with this new version, that incidence won't occur anymore.
Hi, I'm Xing. I'm a first year PhD graduate student. I'm interested in the enzyme called the cellulose synthase. This is an enzyme that is thought to be responsible for producing the cellulose, which is one of the major component of cell wall. So far, we know that in the vascular plants, this enzyme made consists of different subunits. Currently, the priority for me is to test in the non-vascular plants. If not, this complex enzyme complex also consists of different subunits or only one subunit.
Hi, I'm Christina. I'm an undergraduate here. This semester I'm working with the SESA8 gene. So far, Dr. Roberts and I have made more 8KO lines using the G418 vector. We've made GFP SESA8 lines via transformation moss, and those are growing right now. And we've sent tissue from 8KO lines 5B, 6B, and wild type for SFG analysis with some frequency generation analysis. That's what I've been working on this semester.
Hi, I'm Elizabeth. I'm an undergrad here at URI, and this semester I'll be continuing working on complementing the SESA5 knockout with clade A SESAs with their native promoters, and complementing the SESA5 knockout with SESA5 promoters and SESA5, 8, and 3 cDNAs. Also, I will continue immunolabeling protonema and rhizoids of the moss with antibodies to see where the different proteins are expressed in the moss.
Hi, I'm Danielle. I'm also an undergraduate student here. I've been working in the lab since the summer, and I worked with the SESA3 knockouts mostly. I did all the genotyping and the phenotyping. And my results showed that there were no difference between the 3 knockout and the regular moss. So that was interesting to do, but then we found that the moss that I was working with did show the target band. So I have to redo the genotyping and make sure I have moss that does not have the target band. So I'm going to do the genotyping again and then do the phenotyping again to be sure the results were correct.
Hi, I'm Joanna Norris. I'm a faculty member in the laboratory, and I'm working with several of the undergraduates. So most of the things I'm doing are working with different students who are trying to either make a single knockout or a double knockout. And I also have several transformations from double knockouts that were made by a previous student named Marilyn Lee, and I'm going to be genotyping those. So we're trying to make double knockouts where if we haven't seen a phenotype with a single knockout, now we want to make a double knockout with another gene that we think might be related, and see if we get a phenotype with the double knockout.
So as I told you, I would finish up, and you've heard that several of the students have talked about working with me this semester, and I'm going to be working on making some new knockout lines. I'm going to work with MyTran on making some more 6-7 knockouts and some 8 knockouts, and I'm working with Jason to make some new 5 knockouts, and I'm also working with Christina on the 8 knockouts and also expressing some SESA8s in the 8 knockout lines. And another project I'm doing is working with a group in North Carolina at North Carolina State University, and they're trying to figure out what the organization of this protein is in the membrane. In other words, which parts are on the inside of the cell and which parts are on the outside of the cell. And so we're working with them to make some moss that has particular parts of the protein tagged so they can determine whether they're on the inside or the outside.